Good afternoon, 4R5. This is, as promised, the answers to the molecular genetics. I've decided to do a video so that you can also hear my explanation as you check your work. So let's get to it. For the first question, you are given a section of a DNA, all right, which we call the template. So your first task is to find the complementary DNA strand. So by base pairing, T will combine with adenine. Adenine will combine with thymine. Remember, as long as DNA is converted to another DNA molecule, thymine is still present. Unlike when you have the template converted into mRNA. In the mRNA, you do not have the counterpart thymine. Instead, you have uracil. So if you take a look at the mRNA strand and the complementary strand down here, it is exactly the same, except that wherever there is a T, it is now replaced with a U. And likewise, there are several other spots down here. For the second question, the question says, lysozyme is an enzyme consisting of a single polypeptide chain of 129 amino acids. The question is how many or what is the minimum number of nucleotide bases? I need you all to understand that every amino acid is coded by 3 bases. 3 bases which we call codon. 1 codon. Every amino acid coded by 3 bases each, alright, which we call a codon. So if you have 129 amino acids, how many nucleotide bases are you talking about? You have to times 3. For 2B, the focus of this question is over here. Please pay attention that they are talking about an mRNA strand given over here. Alright, so that is why you will see U, which is actually in place of T, thymine. So if you wanted to get the sequence of the DNA, then the T here will actually correspond to your A, will combine with A. And that's why you've got a C, and where there is a U, you will get an A instead, because the U is supposed to be your thymine. For question number 3, it is also similar to question 2B, whereby now you are given the DNA base sequence and you're supposed to find the complementary strand of the mRNA sequence. Wherever there's a T, alright, there is adenine as an mRNA nucleotide. Where there is a C, there will be a G, and where there is a G, there will be a C. Now, boys and girls, for triosine over here, the sequence is actually ATA. Now, you know that A will have a complementary base pairing with a T. But unfortunately, because this is the mRNA, in place of the T, you shall have the U. And I think cysteine is quite self-explanatory. Question 4A is also very similar to question 2B and question number 3 just now. Take note, you are given the mRNA sequence of UAU, and if you are changing it to your DNA, the U is supposed to be a T, and that's why you will see an A over here as a complementary base pairing. Alright, now A, which is found inside your mRNA, will combine with your complementary tyrosine and thymine, and thymine is present in the DNA nucleotide. This is tRNA. You are not required to know this, but then it doesn't really matter. You can just go for it, because the way your DNA, the way mRNA and tRNA communicate is still via complementary base pairing. So where there is a U over here, remember it is a T. And that's why if it's a T, it will complementary base pairing with A. Now, the A over here, on the other hand, is supposed to combine with a thymine. But because this is an RNA strand, that's why you don't have a thymine. And in place of that, you have the uracil. Question 4C, is looking for the 2 ways in which the tRNA differ from mRNA. This is not in your syllabus. You're only required to know the differences between DNA and mRNA, and RNA for that matter. This is another example. Okay, question number 5. You are given the mRNA strand. Okay, and in mRNA strand, you know that the U will actually represent thymine, and if you are asked to convert into the DNA, obviously C will combine with G will combine with C, and so forth and so forth. Wherever there is a T, it will combine with an E. So this is the sequence for your mRNA. Okay, for your tRNA, no worries. Okay, remember it is still by complementary base pairing. Okay, so wherever there is a G, there will be a C here, and it will become a G. But remember, A is supposed to partner with a T, and because it is an RNA molecule, in place of a T, they have uracil. So check your answers. So guys, this complementary base pairing with regards to mRNA, DNA, tRNA, I have explained so many times where so you just need to be careful, okay, where you need to write the T or the uracil. In this question, it shows you the representation of tRNA. Look at this. This is the single strand RNA which fold upon itself using hydrogen bonding. Okay, so that it will expose the anticodon region over here, which has got obviously 3 bases, right? And this is where the amino acid will be attached to. So boys and girls, in this question, okay, I want to highlight to you a few things. Take note, we are working with an mRNA here, and that the mRNA is actually coding for several amino acids. Please be reminded that I've taught you this before. Sometimes several codons may code for the same amino acid. Now the focus is in this part. What happens during mutation number 1? In mutation number 1, you see that everything is the same. Remember, this is not a complementary base pairing. This is really the mRNA, the same strand, but now with 1 mutation. So if you focus over here, boys and girls, the A has now become, okay, that A has now become U. The A has changed to, theoretically, the thiamine. Now, if you take a look at GCA, this is coding for 1 amino acid. Alright, for 1 amino acid. GCA falls here. It codes for alanine. Now, GCU, even though there is a mutation, it also codes for alanine, and therefore there is not much change to the mutation. This mutation is not detrimental to the organism. Alright? For mutation number 2, the mutation actually happens here. Alright? From a U, now it changed to C. So by right, GUU codes for valine, and now that it has become GCU, GCU is over here. So the valine that you were supposed to attach to this amino acid has now been changed to alanine. Now these could be detrimental, alright, to the organism if this is lethal, or if this change in amino acid is actually beneficial, then then the organism will live and survive, and you know, the natural selection and so forth and so forth. Alright. Now, the other thing I want to highlight to you is mutation number 1, mutation number 2, alright, happens at only one particular base. This is called a point mutation. There are several different kinds of point mutation. Okay, but in this case, it is what we call the substitution mutation, whereby one base is changed to another. You have other forms. You have deletion, insertion, inversion. Totally not in your syllabus, alright, and I will not deal with this with you. Okay, I will just jump straight into this particular question over here. You are asked to explain how mutation, okay, may affect the polypeptide for which this section of DNA is part of the code. So for mutation number 1, the point mutation does not change the amino acid, as GCU also codes for alanine. Therefore, the protein polypeptide sequence will not be altered. And for mutation number 2, GUU codes for valine. Because of the point mutation, now it has become GCU, which will now code for another amino acid, alanine. So this mutation has caused the polypeptide to or the protein sequence to be changed.